Yeah, so hello and welcome to Excel Academy. So guys, this is like a small topic which was left as part of the eukaryotic replication, what we were doing yesterday. So I hope you remember like how the different proteins like ORC, ho gaya, CDC6, CTD1, ho gaya, they're coming, they're bringing MCM. Later on, they're getting activated, they're recruiting the polymerase alpha, delta, epsilon, etc. Right? And we were also discussing about the helicase loading, right? So before I proceed, what all we have discussed? So we have discussed that helicase loading, and helicase activation is two different things, right? So we know that once the helicase loading is done in the G1 phase, after that helicase activation is done, and helicase loader will become inactive. Okay, so Akshay, can you tell me how helicase loader becomes inactive once it enters the S phase? Anyone? Tanvi, do you want to try? Yes, Prithvi.
Yeah, CDK actually phosphorylates the CDC6, which is
Yes,
the helicase loader, and then it travels outside the nucleus and goes through the ubiquitin
yes,
pathway.
yes. Very good, very good. So it goes for the proteasome complete protein degradation. Okay, good. Okay, so let's move ahead. So helicase loading, how is this regulation achieved? So just now what we're going to discuss is like how the regulation is achieved. Okay. The cell cycle regulation of CDK activity controls the replication. So already we know this thing. So in the case of Saccharomyces cerevisiae, CDK levels slightly regulate helicase loading and activation. As I mentioned before, many informations about the eukaryotic replication we are getting from the yeast. Okay, we are getting from the Saccharomyces cerevisiae. In the mammalian cells, much of research is still pending. So whatever we have the information about the eukaryotic replication, it is all because of the yeast. Okay. So in case of Saccharomyces cerevisiae, CDK levels tightly regulate the helicase loading and activation. Now during G1 phase, the CDK levels are low. Okay, you can see it over here. So no helicase activation, only helicase loading is happening in the G1 phase, and you don't have CDK levels. It is less. And if what will happen if there is an increase in the CDK level, it is going to phosphorylate your helicase loader, CDC6, CDT1, right? So there won't be any loading. And of course, this is going to result in the helicase activation. Okay. So allowing the CDK levels are low, it allows helicase to be loaded, but the loaded helicase cannot get activated. Okay. Now, during the S phase, elevated CDK activity inhibits new helicase loading and activates previously loaded helicase. So we all know this thing. Now, when a loaded helicase is used for the initiation of replication, it is incorporated into the replication fork and leaves the origin. Okay. You can see it over here. Of G2 phase, M phase, what is happening? You don't have any helicase loading. Why? Because CDK has made them inactive. Okay, is this clear? It is just a repetition, but in a proper manner they have shown, like in the figure, so that it becomes more clear. Simple. G1 phase, helicase loading only, CDK activity is less. Okay, S phase, CDK activity is high, so no loading is happening, but helicase is getting activated. That's all you need to remember as for your exam point of view. Okay. Now, because CDK levels remain high until the end of the mitosis, so no new helicase loading can occur until the chromosome segregation is complete and the daughter cells enter the G1 phase. So if you see the figure also, in the dark, they have shown that the activity level of CDK is remaining high throughout the S phase, G2 phase, and N phase. The moment the new round of cell cycle is happening, that is, it is entering, the daughter cells are when they are entering the G1 phase, then only the CDK level goes down. Okay. So without a new round of helicase loading, reinitiation is impossible. So this is how the regulation is achieved in the case of helicase loading and helicase activation. Okay. And now we are going to just brief about, I'm going to just brief about the finishing of the replication. So remember, I have mentioned that there is a end replication problem. So you have got the 5' and 3'. Your daughter strand is going to be little shorter as compared to your parental strand, right? So this end replication problem we are going to discuss now. Okay. Okay. So the lagging strand synthesis is unable to copy the extreme ends of the linear chromosomes. Now, who can tell me why it is not possible for them to copy the ends of the linear chromosome?
Because there is no place for the primer to initiate. It can't hang on to thin, like it doesn't have anything to hold on to. So that
Yes,
part remains.
yes, very good. So let us just imagine this is your parental upper wala and then the daughter one is there. Okay. So what happens is that when you have during the elongation itself, the end primer is removed. Okay. Ab yahan pe, as she said, which is correct, that you don't have space for, you know, DNA polymerase to come and synthesize, or again, over here you need a primer so that the DNA polymerase over here can come and add the nucleotides. But we don't have the template over here. Okay. And that's why this shortening of end chromosome is happening. Very good. Okay. So not seen during, okay, sorry. The requirement for an RNA primer to initiate all new DNA synthesis creates a dilemma for the replication of the ends. And that's why it is called the end replication problem. In few textbooks, you will see that it is also known as RED. It is a replication end disorder or something like that, they say. Okay. Okay. Not seen during the duplication of the leading strand template. Okay, so this we know that we don't have any problem with this. Now, the requirement for multiple primers to complete the lagging strand synthesis means that a complete copy of its template cannot be made. So, kahan pe problem ho rahi lagging strand mein? So, there was this question where they have asked, CSR ka part C question tha. So, there was no figure given. Okay, so question kya pucha tha? Okay, I don't remember exactly, but I'll pass a message. So what they were asking was like, after the end of the replication, okay, you have got the parental and the daughter. Okay, so two double-stranded DNA is there. Now the question what they were asking is, does the length of the daughter strand is equivalent to the parental strand? That was the first option. So of course not. Second question ye tha, the strands are equal in the length but not complementary. Again, it is not going to make sense because linear application is based on the complementarity. Okay. Third tha that they are not equal. The daughter strands are not equal to the parental strands in the same direction. Okay, same direction मतलब क्या कि यहाँ से छोटा है यहाँ से छोटा है. Okay. Then fourth option was that the daughter strands are not equal to the parental strands in the opposite direction. So guys, what do you think will be the answer? Third or fourth?
Fourth.
Yes, it is the fourth. So it is in the opposite direction. एक यहाँ होगा, एक यहाँ होगा. Yeah, good. So these kinds of questions they are asking, but once you read the options, it is just same sentence, only one word is being flipped. So that's how it becomes complicated. पार्ट से आपको बहुत सिंपल होता है, इट जस्ट दैट थोड़ा सा कॉम्प्लिकेशन आता है इन रीडिंग ओनली. सो इफ यू आर रीडिंग प्रॉपरली द क्वेश्चन देन आप उसका आंसर अच्छे से दे सकते हो. ठीक है. एंड पार्ट बी इज मेनली बेस्ड ऑन योर मेमोरी. सो इफ यू अंडरस्टैंड वेल देन इट इज इजी टू रिमेंबर. मगअप की जरूरत नहीं है, ऐसे बैठ के रट्टा नहीं मारना है, बट वंस यू अंडरस्टैंड देन इट इज इजी. लाइक फॉर एग्जांपल आई विल गिव यू वन मोर एग्जांपल. So DNA topoisomerase 2 is analogous to what in prokaryotes? Gyrase. Simple, right? So essay, if you can compare and study the DNA replication in prokaryotes and eukaryotes, then it becomes easy. So आपको मालूम होना चाहिए कि DNA holoenzyme 3 जो है is analogous to eukaryotes में कौन सा? Yes. प्रोकैरियोट्स में अगर थर्ड है तो यूकैरियोट्स में कौन सा है? DNA all one is your कौन सा? Yes, anyone?
Alpha.
Alpha is a primase. Remember, alpha is a primase. DNA Pol alpha is similar to your DNA G, which is primase. Okay, what about 1 and 3? Epsilon. This is delta. Okay. So there is this table also, which I have shown in the previous lecture. Just go through that once. Okay, no problem. It's okay if you don't remember now, but before your exam, you should be able to know. Okay. So one more thing, I'm gonna share the PDF with you so that you can practice the MCQs. There, I thought those are 100 questions, but there are more than that. You can practice, and they're specifically for DNA replication only. Okay. जो क्वेश्चन नहीं आएगा, just take the screenshot in the mobile and share it in the group so that we can discuss. If suppose you're not able to understand, answers भी दिए हुए हैं, so you can cross check yourself. Okay, so coming back to this. So even if the end of the last RNA primer for the Okazaki fragment synthesis anneals to the final base of the lagging strand template, once this RNA molecule is removed, there will remain a short region which is not replicated, and hence you get the end replication problem. Okay. Now, although this shortening would only occur on the one of the two strands of the daughter molecule, after the next round of replication occurs, both strands of the daughter molecule would be shorter, right? अगर हमने इसको repair नहीं किया, तो क्या होगा? हर एक replication के बाद the daughter molecule, daughter DNA strand is going to be shorter and shorter. Okay. So obviously, this scenario would disrupt the complete propagation of the genetic material from generation to generation. अगर ये चीज रिपेयर नहीं होती तो आज हमारे तक आने तक, like you and me, we would have been looking like a different organism. हम humans नहीं होते, हम कुछ और ही दिखते. And surely the genes which are at the end of the chromosomes would be lost. Okay, so this is the end replication problem. As the lagging strand replication machinery reaches the end of the chromosome, at some point primase no longer has sufficient space to synthesize a new RNA primer, as Shruti said, right? So here you can see in the figure, जैसे जैसे आपका replication खत्म हो रहा है, at the end, what is happening? The last Okazaki fragment, right? So once you remove this, what will happen? There is a space present, gap is there. Now, if I keep on replicating with this, can you see it over here? Eventually, the chromosome is becoming shorter. Can you see? Gray वाला तो आपको full देगा. But then what will happen? The next one, you will see that ये जो है छोटा, the next round it is going to be the small only. So you can see that this much short it is as compared to your parental one. Can you see it? Okay.
Yes, ma'am.
So this results in the incomplete replication and a short single-stranded DNA region at the 3' end of the lagging strand DNA product. Now when this DNA is replicated in the second round, it is going to be shortened. Same thing, it's just that it is the description of the figure. Okay, now how to solve this? So this is one mechanism, and the second mechanism, which you all know, is of telomerase. So let's talk about the first mechanism. So one solution is to use a protein instead of an RNA as a primer for the last Okazaki fragment at each end of the chromosome. So now what we are saying is that till now we were using RNA primer, right? So अब हम RNA के जगह protein use करेंगे. So this is first mechanism, okay, to use protein. Now, in this situation, the priming protein, now it is known as priming protein, it binds to the lagging strand template and uses an amino acid to provide an O wedge. और हमें क्या चाहिए था, OH ही चाहिए था. Remember in the primer also we have got 3-OH, to which your incoming phosphate comes and bind and you have got the formation of a phosphodiester bond. Remember the SN2 mechanism we have talked about. Remember the first few lectures. Okay, so typically this OH is now, instead of the ribose of the ribonucleotide, it is now provided by the amino acid. Okay, and which amino acid has got the OH? Of all the 20 amino acids, how many amino acids are there which has got the OH? Yes.
Three.
What are those names?
Tyrosine,
Yes.
threonine,
Yes.
and serine.
Yes. Very good. So among all these three, we are using tyrosine. Okay. Now, serine and threonine, why we are not using it? See, it again depends on the environment in which it is present. Okay, so mainly whenever we talk about like DNA replication, anything which is related to the DNA and all, you will see that maximum time tyrosine has been used. If you remember in the previous also, when we were talking about the fingers of the DNA polymerase. So in order to understand, in order to make sure that the base stacking is proper, again, the tyrosine was involved. Okay. This is actually not written anyway. This is my general observation that whenever we are talking about DNA polymerase, whenever we are talking something related to the DNA RNA, I have noticed that the maximum enzymes in their catalytic site or whatever, they have got tyrosine present. Okay. जब हम proteases के बारे में पढ़ेंगे, like different types of proteases are there or anything which helps in the breaking of the product and all, you will see that maximum time over there histidine is there. ऐसा मेरा general observation है, it's not written anywhere. Maybe you can also observe next time whenever you talk about any type of protein. Okay, this is just my observation. Okay, so we have got three types of amino acid which can provide OH group, of which tyrosine is typically present. Okay, so this replaces the requirement of the 3-OH which is provided by the RNA primer. Upon आपका priming protein है which is giving the OH. Now to this OH, okay, to this OH it is going to bind. So by priming the last lagging strand, the priming protein becomes covalently linked to the 5' end of the chromosome. Terminally attached replication proteins of this kind are found at the end of the linear chromosomes of certain species of bacterial and animal viruses. Okay. Okay, so let's see. In the figure, it will be more clear. So can you see it? We have got the problem over here. So what is happening? Then the protein is coming. This protein, we have attached it over here. Can you see it? The protein, priming protein. Okay. So now this OH is being provided by this and the polymerase is going to synthesize. Are you able to imagine कैसे हो रहा है? Yes?
Yes, ma'am.
Yeah. So it's just that it goes and attached to the 5' end. Okay. But it is providing, it is just, इसे expose कर दिया है इसने OH group को. उस OH group को use करते हुए DNA polymerase ने आगे synthesize कर दिया है. ठीक है? It is not that वो यहाँ आके बैठा है. Just one second. Yeah. So you can see that at the 5' end it is there. Now this protein will have exposed OH group. Okay, and then they will start synthesizing. So for longer DNA molecules, this method combines with the conventional origin function to replicate the chromosome. अगर जैसे कुछ ज़्यादा लंबा करना है, the gap is not being filled or whatever it is, then you can have this thing. Okay. So this is one method. Clear? Okay. Now
Yes,
one
ma'am.
more thing here you need to understand is that in this case, the normal replication is not happening. Can you see it? It is not that the only end is being replicated. The entire chromosome is being replicated. Okay. Instead of having our RNA primer, they initially itself, before replication itself, they have started using the protein priming and replicated the entire chromosome. This is happening only in few cases of bacteria or in few cases of viruses, those who have got the those have got the linear DNA. ये एक तरीका है to avoid end problem. Okay. The second method, let me see just, yeah. The second solution is to have the role of telomerase. Is it clear, the first one? See, till now we talked about how prokaryotic replication is happening in a circular chromosome, right? Then we talked about how eukaryotic linear DNA is getting replicated. Now, in few cases like viruses, जहाँ पे आपके पास, you have got the linear DNA, okay, and they don't have telomerase. So, in order to avoid the shortening of the DNA, they might use this protein priming to start. Instead of having RNA primer, they're going to have a protein primer and they're replicating the entire chromosome. It's not that the only end of the chromosome was replicated. It was the entire chromosome replicated. Okay. Is it clear? But this is rare. Okay. This is rare. They have not given any example also in the textbook also. And even I have searched like where this is happening. They have just seen in few viruses, but they are not sure about it. So that's why it's just given as a small topic. Okay, it's not completely say verified or scientifically shown. Okay, so in the case of the eukaryotes, we know that they have got the telomeres. Okay, and telomeres we know that why even though they are not contributing to any gene expression, but they are important. Now, why telomeres are important? Can anyone tell me the importance of having a telomere, even though they don't contain any gene expressing, for the or any important genes for proteins or mRNA expression, but still they are important. The shortening of telomeres results in so many genetic disorders. So, can anyone tell me why?
The longer the telomeres, the more time the cell has before important genetic material is stripped away. So if the telomeres are already short, then you start losing genes a lot faster.
Okay, this is one good reason. Then what else? What else you can say? This is the same thing you can tell in the other language, in other wording also. Yes.
It prevents the chromosome from sticking to each other,
Yes.
to remain separate.
Yes, yes. So it makes sure that the telomeres, the chromosomes doesn't fuse together. Okay, even though you will see that the telomeres is getting fused, this is only happening when the telomeres are short. Okay, if suppose I have got a good amount of telomeres, then it is not going to stick to the other chromosome. If suppose I have got short telomere, then there is chance that it is going to fuse with the another chromosome. Now, fusing with the another chromosome means what? They might be having the overhangs which was complementary to each other, and then only the 2 or 3 chromosomes are getting fused to each other. आया समझ में?
Yes, ma'am.
हाँ.
Yes.
Okay. So, okay. So, for example, in the case of human telomeres, it mainly consists of head to tail, the sequence of 5' to TTA triple G. ठीक है, अगर हो सके तो please remember the sequence, because you really need to remember this, because once you remember the sequence, then how exactly the telomerase is functioning becomes easy. कुछ ज्यादा नहीं है, छोटा सा ही है. 6 nucleotides are there, TTA triple G, बस आपको उतना याद रखना है. Of course, please remember the direction also, 5' to 3', TTA triple G ऐसे याद रखना है, because question में आपको 3' to 5' भी दे सकते हैं, just to confuse, and they will give you as triple G ATT. Okay, so this completely looks different to this. हमने कैसे याद किया है TTA triple G? अभी ये दे देंगे तो हमें अचानक से click नहीं होगा. But if you see the direction and read it like this, it is the same. Okay, so mole bio में ये थोड़ा सा trick किया जाता है in the questions, just to confuse the child. Okay, तो थोड़ा सा ध्यान देना whenever we talk about, and wherever I'm mentioning कि ये sequence याद करना है, so please guys, okay, please do remember those sequences. Okay. Oh. Yeah. So although many of these repeats are double-stranded, the third dash end of the each chromosome extends beyond the 5' end. क्यों? Because of the telomerase. And वो कुछ नहीं है, बस TTA triple G के repeats हैं. Okay, so that's why it is possible. So that's why it is possible for the telomeres, to protect the end of the DNA. Okay. Okay, so telomeric replication एक छोटा सा intro दिया है, but like maybe not tomorrow, or maybe on Sunday, we are going to study about the telomeres and telomerase in details. Okay, I'm just giving you introduction now. So the ends of the linear chromosomes cannot be fully replicated by the semi-discontinuous replication, as there is no DNA to elongate to replace the RNA, which is removed from the 5' end of the lagging strand. Okay, so this could be lost, and that's why we have got the overhang. ये आपको मिलेगा overhang. ठीक है? This is the overhang. Now, what do you see over here? After the telomerase has repeated sequence, TTA triple G, N time. So those many repeats you are going to see once the telomerase function. Okay. Now, what is telomerase? It contains a short RNA molecule, whose sequence is complementary to this repeat. Okay. So this RNA is going to act as the template, and it is going to help in the synthesis of the DNA. So telomerase क्या हो गया फिर? If I say that telomerase is a kind of DNA polymerase, is it true or false? Yes? Yes? Anyone?
No.
The answer is true. Why? Just now I said that it is having the RNA template, okay, and it is going to synthesize the DNA. So synthesis of DNA matlab kya hua? It is a DNA polymerase, okay. But whenever we talk about RNA polymerase, we always mention that it is a RNA dependent DNA polymerase. Okay, so ये भी important हो गया याद करने के लिए. You will see. I'll show you the diagram, then it will become more clear. So this RNA acts as the template for the addition of the repeats, and then finally what you get is the overhang. Okay. Okay. I'll show you the figure. Yes. This is the very basic figure how a telomerase is functioning. Okay. And this is not what they are going to ask in the exam. This is just for your understanding, first time understanding. Okay, so let's see. So what you can see in the figure is that you're going to have an at the 5' end of the daughter strand. Can you see it? At the end of the 5' daughter strand, you're going to have a gap. क्योंकि हमने अभी RNA हटा दिया है. Okay. Now what is happening? The telomerase is coming. This telomerase is coming, and this portion of the telomerase is of RNA. Okay, ये RNA है, RNA template है. Can you see U over here instead of T? And
Yes,
what is
ma'am.
the sequence over here? Can you see the sequence? It is AAU and triple C. And what is the repeat we have? It is TTA triple G. Correct?
Complementary.
Yes, ma'am.
Yeah. U के अगेंस्ट में हमें A आएगा. यहाँ पे T है, यहाँ पे T है. Correct? C का G. Now, what then? Okay, so we have got the complementary RNA template over here. Now what it is going to do? See, you have got the template over here, correct? यहाँ पे हमारे पास template है. Now you're going to add the nucleotides. So अभी क्या हुआ है? इस direction में you have added more amount of DNA synthesis. Now the question is coming, okay? छोटा तो हमारा ये वाला स्ट्रैंड था, डॉटर स्ट्रैंड. हमने पेरेंटल स्ट्रैंड्स को क्यों लंबा कर दिया? Yes.
Because the parental strand is used by the lagging strand is made by the normal process itself. So in order to create the template for the lagging strand, they increase the parental strand.
Yes, very good. So telomerase क्या कर रहा है? So see, can you see it over here? The 5' end में gap था. Okay, so obviously the parental strand was 3' end. है ना? So we can add easily over here the new nucleotides with the help of the telomerase. So telomerase have added so many nucleotides over here. Okay. Now using this, now I have the template. Now using this, the DNA polymerase is coming and RNA primer डाला है, उसके बाद उसने add कर दिया है, and finally the ligase will come and seal the nick which is present. Okay, like how normally Okazaki fragment is being made. Okay, so obviously this DNA polymerase is going to be your epsilon. ठीक है, clear? इतना clear
Yes,
है
ma'am.
सभी को?
Yes.
Okay. Yeah, okay, so only that much was pending. And yes, so tomorrow, not tomorrow. Wait, just let me stop the recording.